The senses of the ear, that would be hearing, balance, and the ability to perceive acceleration in both the linear and vertical planes, these senses are perceived by epithelial cells known as hair cells. They are known as hair cells because on their apical surface they possess an enlarged cilium known as the kinocilium and specialized microvilli known as stereocilia. Movements of the fluid of the ear, such as endolymph, can cause these hairs to move, or in the case of the vestibule and the semicircular canals, this fluid can then move the gel that these hairs are immersed in. When the hairs bend, this causes proteins which attach the tips of each of these stereocilia. This causes these proteins to pull on ion channels which are mechanically gated. So as one stereocilium moves, it then pulls on the mechanically gated channels of the neighboring stereocilium, causing ions to enter, which affects the electric potential of the cell. As the cell depolarizes, this then changes the activity of the voltage regulated calcium gates, which control the exocytosis of neurotransmitter. So the movement of the hair cells will affect the electrical potential of the cell, which then determines how much neurotransmitter these epithelial cells release onto the sensory neurons of the vestibulocochlear nerve. The vestibulocochlear nerve then carries information to the brain about sound, balance, and acceleration. The hair cells of the cochlea detect sound. The hair cells of the semicircular canals detect balance. The hair cells in the saccule and utricle of the vestibule allow us to perceive acceleration. The hair cells of the utricle allow us to perceive acceleration in a linear plane, while the hair cells of the saccule allow us to perceive acceleration in a vertical plane.